Um, Hello everyone and welcome to my talk. Just a quick outline before we get started. When I speak about a loris, I'm referring to a slender loris, as you can see on the left. When I talk about Nycticebus, I'm talking about that fluffy little guy in the middle, the slow loris, and Periodicticus refers to that pothole on the right. These figures will be used throughout the presentation for anybody who's less familiar with Strepsirrhines. Strepsirrhines are a specialised primate clade. They include the modern lemurs of Madagascar and the lorises, potholes and galagos of continental Africa and Asia. Now, among the extinct relatives, there are the Adapiforms, as you can see on the left. They're usually regarded as stem Strepsirrhines, and a series of taxa more related to the modern lineages. Now, lorisoids, where we are going to focus, are divided into two families, one comprised of potholes and lorises, and the other comprised of galagos. The map on the right there shows you the biogeographic distribution of lorisoids in several shades of grey, and the little black stars mark fossil discovery sites. Now, much is known about the modern lorisoids, but this is sadly in stark contrast to their fossil relatives. This is due to a paucity in the fossil record. The aim of this study is to deduce the sensory capabilities of the early Miocene lorisid Mioeuoticus shipmani. The timing of the evolution of the characteristics in this animal has important phyletic position implications for it and its modern counterparts, but more importantly, for reconstructing its behaviours. Now, the specimen we use is a holotype. It's housed in the National Museums of Kenya in Nairobi, and we're using the sole cranial CT scan rendered in Aviso 3D. So first, I study the olfactory system. These are known as turbinals. They are a network of spongy bones in the nasal cavities. You have them too. We process smell this way. There's two nasal cavities separated by the nasal wall right up there in the middle. And as you can see, in each of the nasal cavities, there's a symmetrical pair of turbinals. I've color-coded them here. In red, yellow, pink, and purple are the maxillo, naso, fronto, and interturbinals. There's singular pairs. And then in three pairs, you have green, blue, and orange, which are your ethmoturbinals. The little brown fragments at the back we think might be a fourth pair, but sadly they're not diagnostic. You can see that there's not much left there. There's likely much better preservation than what we're looking at here, but sadly the scan resolution we're working with was not great. So just to show you how poor, on the right, this is a modern Periodicticus pothole. You can see those beautiful little white scroll-like features inside. They're very, very ephemeral. On the left, that's Mioeuoticus. See if you can see any of them in the Mioeuoticus scan. And if you do this for a couple more months, then you probably get the result that we did. Next, I reconstruct the bony labyrinth. I'm going to measure the size and the orientations of the semicircular canals in the cochlea for different comparisons with fossil and modern primates. And I want to try and deduce mode of locomotion and positional behaviour. For those who aren't familiar with the anatomy of the inner ear, because I wasn't, I will give a quick run through. The modern Periodicticus example is on the right there. You have three semicircular canals, anterior, posterior, and lateral. Common crus is the little stick-like feature which separates the anterior and the posterior. The canals then run down to a vestibule. They run through to a utricle and down to the cochlea, which has that famous snail-like feature. The angle between each of these canals is known as the angle of orthogonality. So you can see that with these little red angles here. Now, all of these features are distinct. The structures are not observable completely, sadly, in the Mioeuoticus on the left. Again, the resolution of the scan is not great, but I could get general morphology and I could get that angle of orthogonality. Next, I study visual acuity. So I measure the interorbital space, which is, first of all, the optic foramen area, which is where your optic nerve protrudes straight through the skull. And then I take the orbital area, which is in red, which is where your eye fits nicely into the socket. And we enter this into the optic foramen index equation, which was made famous by Kay and Kirk in 2004. Now, I won't bog you down in the maths. There's a lot of it. I'll keep this short. If you want a more in-depth description, you can read our recent publication. Yay. Now, basically, the result is submitted into two larger optic foramen quotient equations. They numerically define the visual acuity of any primate, whether it is considered diurnal or nocturnal. This results directly compared to other fossil and modern primates to tell you how this animal could see. Now, looking back at the terminals, if you imagine that this black line is the nasal wall straight down the middle, and you're looking into a single nasal cavity of a lemur on the left and a loris on the right, most striking is the location of the interterminal, which is in purple. So in these animals, compared to Mioeuoticus, it's really interesting. In lemuriforms, as you can see on the left, ethmoterminal 2, which is green, originates from ethmoterminal 1, so they connect. They are shared. So the interterminal in purple sits between the shared ethmoterminal 1 and 2 in yellow and green and ethmoterminal 3 in blue. As you can see in lorisoids, however, this green second ethmoterminal directly connects to the nasal wall. So this little purple interterminal sits between ethmoterminal 2 and 3. In Mioeuoticus, you can see it looks exactly like that of lemuriforms anteriorly. But as you move further back, it actually looks much more like the lorisoids, except for that little interterminal has moved. This is completely unique. It now sits between ethmoterminal 1 and 2, yellow and green, rather than 2 and 3, green and blue. Looking into the olfactory terminal surface area overall, the graph on the left shows you total olfactory terminal surface area relative to cranial length. Now, you have your blue shaded section, which is your haplorhines, which is like your monkeys and your apes. And then you have your red shaded section, which are your Strepsirrhines, your lorises, your pottles, and your galagos. Now, you can see Mioeuoticus in green. It's just below that red shaded section. Now, the terminals were not complete. There was taphonomic loss. Also, you could see the scan wasn't at its best. I do believe if we had a full terminal assemblage, it would sit just nicely inside that red shaded area. But as for where it does sit, the overall surface area is very similar to Propithecus verixi, which is the little guy on the left, Mioeuoticus elegantilis in the middle there, and once again, Paradicticus potto. The large olfactory recess length that it has, this total expenditure in its nose, is considered a keen sense of smell. And also we found a vomeronasal groove, which is a feature used in hormonal scent attraction. Looking at the inner ear, there is a 105-degree angle between the semicircular canals in Mioeuoticus. Now this indicates a slower rotational head speed and a lower mean vestibular sensitivity to motion. So kind of imagine him moving a bit like a sloth. Mioeuoticus cochlea, it is broad, it's bulky, similar to the loris, which is in the top left. And it's much larger than Nycticebus in the bottom left or Paradicticus in the top right. The common crura is short relative to the cochlear and semicircular canals, with a narrow utricle and vestibule. This is very similar to the Nycticebus in the bottom left. And the strong ovate shape that you can see in the semicircular canals is comparable between Paradicticus and Mioeuoticus in the top and bottom right, but the canals are morphologically closer to Nycticebus in the bottom left. Now, the OFQ value that we got was 16.91. It's very similar to extant diurnal Strepsirrhini Hapalema griseus in the top left. Yes, it's diurnal, but it only comes out at dusk. And the nocturnal Galagomachi in the middle, and again, Paradicticus potto on the right. So the Mioeuoticus is more consistent with a nocturnal activity pattern. Taking into consideration, it's suggested to be a lorisid. This OFQ value suggests visual acuity adapted to low light levels. Now, the modern lorisid behavior is nocturnal. It's olfaction orientated when compared to monkeys who are more vision orientated, and they're slow moving. Slow moving is really weird among primates. The rest of primates, relatively fast moving mammals. Mioeuoticus did behave quite similar to modern pottos, meaning this really strange, specific type of behavior is quite ancestral for the family. And we believe this particular type of behavior came from their very origins. So we suggest Paradicticus potto, which is still came up time and time again, has retained a primitive optic morphology as a plesiomorphic character inherited from Mioeuoticus, and future research would benefit from studying the sensory capabilities and behavior of this animal as a living model for lorisid evolution. So I'd like to thank everyone on this list. I'd like to thank you all for listening, and in particular here to Jakub Zalewski of the University of Warsaw for this beautiful paleo art reconstruction of Mioeuoticus in its natural environment. Thanks.